So this is Reactome's homepage. You'll see that I've put the URL at the top of the page, nice and easy to remember, www.reactome.org. Nothing very remarkable about it. We've grouped perhaps the most important and useful sections into this group of 6 buttons in the sort of top left-hand corner. The other thing that's not so easy to spot is the search function. So the search function is this on the right hand side of the menu, and the search will allow you to type in the words of molecules, the names of molecules as a word, or a collection of words if you're looking for a pathway. It also recognizes UniProt and KEBI identifiers, and in fact identifiers from a lot of other databases as well. So when you type in a phrase of something you're looking for, it uses predictive text to try and work out what you were looking for, and it will make a guess about what the word is that you're trying to type in. So you'll see here I'm typing in the word calmodulin, and it's guessed correctly that that was what I was looking for. If it guesses correctly, you can select the name from the list that it provides, or if it doesn't, you can click on the search button. And the results will come back, and it will look something like this. So we have the results are split up into sections of complexes, reactions, pathways, regulation, sets, and proteins. So it tries to be intelligent about the order in which it represents those sets. It thought that calmodulin, it was most likely a complex, so it's showing the complex results first. Within each set, it gives you the top 5 results, but there may well be more than 5 matches, and you need to click on the word that's in the header of the set in order to see the other hits. You can see that the reason it chose that complex here is because it contains the word calmodulin, and it's highlighted the matching word in grey. If I'd used 2 words and it found 2 of them, it would highlight them both. You have some filters on the left-hand side, so you can filter your results by the species, by the type, by the compartments that the molecules occur in, and in fact by reaction types. So, but I should point out that reaction types is currently a little bit hit and miss whether the filtering works correctly. But so any of these results, when you've decided that it's something you are interested in, if you click on any of those names, it will take you to a panel of results for that section. So I've chosen this example because I wanted to show you. It looks a little strange here that when I select calmodulin as a protein, I've got 3 identical matches for CALM1, so CALM1 being the gene name for this protein. So why do I have 3 hits here? Well, if I move on to show you the next slide, I've picked the top one from that list of 3, and now I can see the result for just that one hit. I can see that it has a compartment term of plasma membrane. And I can also see that I've got other forms of this molecule. It's showing me that we've got CALM1 in the cytosol and the extracellular region and the nucleoplasm. That's the reason for the multiple hits, because we consider that calmodulin in the cytosol is not the same thing as calmodulin in the extracellular region or the nucleoplasm. So sometimes if you've searched for the term that will match the protein wherever it is in the cell, you can get multiple hits. So anyway, so this particular version of it is in the plasma membrane. The other thing to notice here is this section called Locations in the Pathway Browser. So this is trying to show you where you can find this molecule in pathway diagrams within the database. But by default, this is a hierarchical view where the hierarchies are hidden from you. And if you want to see the full list of the hierarchy, you need to click on the plus symbol on the left-hand side, which will then expand the hierarchy. There we go. This is what it would look like. So you can see now I've expanded the hierarchy, and I can see sub-levels of that hierarchy representing subpathways, and finally complexes and individual molecules. And so this will allow you to go to specific events or even specific molecules within the pathway diagrams. And again, any one of these names, when you click on it, will take you to open the pathway browser and show you a pathway diagram. But it's very important that you remember that those locations are, by default, they're currently hidden, and you need to expand in order to see all the places you can find this molecule. Okay, so this is what the pathway browser looks like. It's essentially divided into three sections. We have on the left-hand side a hierarchical organization of all of our pathways in a panel called the Hierarchy panel. This works very much like the Windows file manager. The little plus symbols along the left-hand side, if you click on them, will open up the lower levels, the sub-pathway levels of our organization. The top right panel here we call the pathway panel, or the overview panel. At the moment it's showing you an overview of all the pathways in Reactome. And at the bottom right hand side we have what we call the detail panel. So these panels are all connected. If you click on something in the hierarchy panel, you will see that highlighting take place in the hierarchy and in the pathway panel and in the detail. The details will change to reveal the details of the thing you just clicked on. Other things to notice is this button at the top here, the tour, video tour button. There's a very nice video tour through the database with a lot more detail than I'm going to cover today included. There's also this layout collection of buttons which will open and close these three panels depending on, to make more space for the one that you're interested in. There are some zoom tools here which allow you to move around within diagrams and to zoom in and out. If you have a mouse connected to your computer, then you can also zoom using the scroll wheel, and you can click and drag the diagrams to move them around, which is probably easier than using these buttons. Okay, so looking at the pathway overview, you can see that we have these collections of what look like concentric rings of dots. So the way that works is the central point, the central node, is essentially the highest level of organization within that topic within Reactome. So you can see, I hope, that this one here is metabolism. So this node in the center represents the entire topic of metabolism, and then around it is a concentric ring of nodes that represents sub-pathways of metabolism, and then around that is another ring of sub-pathways, and so on out to the farthest reaches. So what we have is a representation, a radial representation of the sub-pathway organization of that topic in Reactome. Sometimes you'll notice that there are arcs that join the concentric collections of concentric rings. That's because there are some topics that occur in more than one place within Reactome. So here we have the topic of hexose transport, which is, because it involves hexose, it's part of metabolism, but because it's transport, it's also relevant to the topic of transmembrane transport. So you will find that these arcs are representing essentially pathways that have been used in more than one place. So if you remember earlier I said reactions can be reused in more than one pathway, and in fact sub-pathways can be used in more than one pathway topic. Yes. Within the hierarchy panel, that left-hand section, you will see that there are these icons, and these icons have a key. I'm showing on the right-hand side. They mean something. The box, the set of boxes connected by lines tells you that that object is a pathway. The two boxes going to one box with an arrowhead represents a reaction. This two boxes joined by a broken arrow is a black box event, which I'll explain in a minute. The double greater than sign means that that event has been inferred from a model organism. So if you remember, this is where there's no human experimental data available and we've needed to use experimental data from another organism. And then there are icons to tell you when a pathway is new, when it's been updated, and when it represents a disease process. So our pathway diagrams actually use something called systems biology graphical notation. So we didn't invent this iconography. We try and use standards wherever we can. And what that means is that small molecules are represented as ovals. Green boxes are proteins. Blue boxes are complexes, and blue boxes with a double boundary around them are sets. So sets is something that confuses people sometimes. Within Reactome, if we use a set, what we mean is that the molecules within that set are functionally equivalent to one another. Any one of those things, if present, is sufficient for the reaction to take place. So set means any one of those things is what's required. Very different from a complex. For a complex, that represents the need for several proteins bound to one another in order for the event to take place. If you remember, here is a simple reaction, and we can see we have input molecules connected by plain lines to a central reaction node. We have outputs connected to the reaction node, but there are arrowheads at the end of the lines to tell you the direction, so we know these are the outputs of the event. We have a catalyst connected to the reaction node with a line with a circle on the end, so we know this is the catalyst. And in fact, within the diagrams, we show things within their correct compartment of the cell, and the compartments are represented as a pale pink box with a double orange boundary line around it. So this L-shaped line here, I'm only showing you the corner of the compartment, so that tells you that this is the boundary membrane around that compartment. So this molecule AA is sitting on the membrane of that compartment. We can show regulation, so here we have an arrow representing a positive regulator. I know it's positive because it has an open arrowhead on the end, and this is a negative regulator, which is shown by a sort of a T shape at the end of the line pointing towards the reactome node. And there are actually several different kinds of reaction node. So the default one is the open square for a transition, which essentially means something happened, it's not one of the other classes. A very common class is binding, which is represented with a filled-in circle. The opposite of that is dissociation, which is a double boundaried circle. And then we have two more, which both would be classed as black box. Let's start with the easier to understand one, which is uncertain. So if we represent an event in a reactome where we know some of the molecular details, but we think that the process may be more complicated than we're able to show, then we'll suggest it's an uncertain process. A good example of that would be something like a kinase cascade. So you can do an experiment where you introduce an excess of a kinase X and you show that protein Y becomes phosphorylated, but if there's a suspicion that there's an intermediate step, so the kinase is not directly phosphorylating Y, it's actually it's phosphorylating another kinase, which is then the kinase responsible for phosphorylating Y. Under those circumstances, we would show that as an uncertain event. In other words, there's some doubt about the molecular mechanics of it. Then we would use this node type for uncertain. The remaining one, the emitted process, this is very different. This is used where we have an event where we do know what the molecular details are, but we've simply left them out to make space, to save filling up a diagram with lots of unnecessary detail. And the classic example of that within Reactome is to show transcription and translation as the result of a transcription factor binding event. So what you would find instead is you would have the transcription factor migrating to the nucleus, and then the next event would be the magical appearance of the protein. So we miss out the binding to the gene promoter, we miss out the transcription and translation. We would just show you the final outcome of that, which is the process becomes available within the cell to continue the signaling. So if you wanted to know the details of transcription and translation, they are available elsewhere in Reactome. So we effectively leave it out to make room for the other things which are more relevant in the context of the pathway. Navigating in the pathway browser, as I said, everything within the hierarchy is linked to what you see in the pathway diagram. If I select a pathway name, it will highlight it in the diagram. In either case, whichever way around I do it, the details of that event will appear in the detail view. For some pathways, when you open them, sub-pathways within that are highlighted using colored boxes to show you the groupings of events, to give you a visual clue that what's contained within this blue box here is a sub-pathway of the whole, and there's a different sub-pathway over here represented in the green box. If I am looking at a pathway diagram and I select a subsection of it, so here I've selected activated notch transmits and so on, that highlights that set of events with this dark blue color. The yellow coloring over here is where I've moved my mouse pointer over another event. I haven't actually clicked on it yet, but because I'm hovering my mouse over that event, it's highlighting it for me in a different color to show me that it's distinct from the thing I've already selected. But notice that the details panel won't actually update until you select something. So highlighting is not the same as selecting. When I'm zooming in and out of the pathway browser, when I open a diagram, it typically will open something like this. So you're seeing the whole pathway. You may or may not get sub-pathways highlighted within these colored boxes. But as I zoom in a little, it will show me more and more information as I carry on zooming in. So I zoom in and I start to be able to read the labels in the boxes. If I zoom in a bit more, I can see trivial small molecules beginning to appear because they're hidden in the zoomed out view, they're hidden to make the diagrams tidier, but as you zoom in they start to appear. You also see these small red icons which tell you about the availability of molecular interactions. And if I zoom in to the fullest extent, then the objects will start to reveal structural details. So here I've zoomed into a protein and it's showing me a protein structure, and then above it I hope you can see this small molecule is also revealing a chemical structure. Yes, and then I've zoomed into so you can see that a bit more clearly. Okay, also within the diagrams we have something called the contextual information panel. So if you hover over an object on a diagram, it will grow one of these little arrows on the right hand side. If you click on that, it opens a panel and that will show you the molecules contained within the object. It will give you a link to any other pathways that molecule participates in, and if it has interactors, it will show you a panel of interactor information as well. I said just now the little red icons with a number in are showing you that this molecule has molecular interactors available in the default database. And the default for Reactome is the intact database. So the number of course tells you how many interactors are available. If you click on it, it will then show you a radially laid out set of those proteins. If you click on the line, the edge that connects the interactor to the original object, that will then open a page of information from Intacct to show you the evidence for that interaction. There's also a slider panel here where you can filter by the confidence score to show more or less of these interactors depending on how confident they are, depending on the values that we get from Intacct. You can download the interactors for the whole of this pathway by clicking on this little button that looks like a cloud with an arrow on it. You can also search pathway diagrams. This is a separate search from the one that's available on the home page. If I type in the name of a molecule, it will try and find it within the diagram. If it finds it as more than one object, it will give me a list of them, and if I click on one of those, it will tell me this molecule exists directly in this diagram. Part of these complexes, it participates in these reactions, and it has these interactors, and all of these are links which, if you click on them, will highlight that thing within the diagram that you're looking at. Okay, we also have a way of changing the color schemes if you want to. There's a settings panel on the extreme right hand edge, which if I open it up and change from the standard color scheme, so I think the one I was showing you before was called the modern color scheme. This is the standard color scheme, which is and so you can change the way the diagrams look. If you find that you're being confused by what all the objects are, there is a diagram key which is represented up in the top right hand corner of the pathway diagram, and you can see it covers all of the commonly found objects within diagrams. If you want to export a diagram, you can click on the little camera button on the top right hand corner, and that allows you to download the image as a reusable graphic. And for some pathways, not all but some pathways, you have this extra button of illustration, and if there is an illustration available, clicking on that will open up these rather nice graphics, illustrations of the pathway process. Okay, so the details panel, the bottom right hand section, I haven't really told you what to expect to find in that section, but essentially it's details of whatever it was you'd selected. The important things to know about are the background information is represented as a summation. The inputs and outputs are represented, and you can select these inputs and outputs by clicking on the buttons on the left-hand side. Very often you will find there's more information if you reveal that information by clicking on the plus symbols on the right-hand side. So if you remember, left is select, but right is reveal. That's worth remembering. The key literature, the literature that contains the experiments that prove that this event takes place, is in a section called References. So you can sometimes find references in more than one place in the details panel. You can find them under summation sometimes, but those aren't the key references. The key ones are in this section called Reference. Okay, when I'm looking at the details panel, there's the overview, which we just looked at, but there are several other tabs. So there's a tab called Structures, and this is where you look to see whether any of the molecules that you currently have selected have a structure available in the PDB database. If you have a chemical selected, then it will fetch information from the KEBI database, and in fact if you have a reaction selected, it will try and find information from the RIA database. If you look on the Expression tab, it will fetch expression information from the Expression Atlas database and show you a table and small graphic of the proteins in the object that you currently had selected. So you can see here it's trying to give you an idea of the expression of these proteins within normalized human body tissues, body parts. This information is coming to us from the Expression Atlas, they've done the clever part of picking what they consider to be the normalized expression data sets. And then the final tab on here that I'm going to show you is the Downloads tab, and this is where you can select to download the entire pathway in a number of formats for reuse. Okay, so disease information in Reactome, I already told you that we now do disease pathways. It doesn't really differ that much from a normal pathway view. So you would click on the name of the disease pathway, and the disease reactions would appear overlaid on top of the normal pathway equivalents, which are shown as sort of grayed out in the background. And if you want to know what diseases are significant for this event, then there's a section in the details panel actually called disease. So that's how you find that out. So the interpretation of high throughput data, the Reactome tools, let's very quickly take a look at that. So the way you get to these tools is either from the home page, you click on the Analyze Data button, or if you go into the Pathway browser and click on the Analysis button, it does exactly the same thing. It opens up this analysis submission panel. It's a common panel for all the types of analysis. It's expecting one of two formats. This is the first one, which essentially is just a list of identifiers, one per line. It recognizes lots of different identifiers, but the ideal ones to use are UniProt IDs. Gene symbols work very well too. So you input your list, or you browse to a file, you click on Continue. You then have a couple of options. You have Project to Human. So by default, if you give us a list of identifiers and some or all of them are not human identifiers, we will try and find the equivalent human identifier, because the statistics work best if you've given us human identifiers to use. There's also another option of Include Interactors. So that's not the default. By default, you will only be searching molecules which are actually in the Reactome database. But if you choose to, you can also look at the intact interactors of all of the things within the Reactome pathway. Of course, that greatly expands the space that you can that your experiment is searching against. But you are then going into interactors away from the curated information and into experimental interactions. And the result comes back and it will look like this. It's superimposed into the Pathway browser. The table of results appears in the detailed view. It's sorted by probability score. So here it's a corrected probability score and false discovery rate corrected. As you click on the name of the pathway, it will highlight and open it in the Pathway browser. It will also show the highlighting and the hierarchy. Okay, in the interest of time, I'll go very rapidly through that. You're also looking at some scores for pathway topology. So here, rather than entities found, we're now looking at reactions found. So this is also represented in the hierarchy as an orange box around the name of the event that's been matched. This is simply another way of looking at the results. So here, instead of just treating the pathway as a set of molecules, which can be matched by the set of molecules that represents your query and looking at the probability of the overlap, now you're looking at which events have been matched, where are they in the pathway, and how many of them have been matched. So it's you can spot things such as all your identifiers have matched the beginning of a pathway that might mean something that you don't get from overrepresentation analysis. Okay, oh, and you can download the analysis results as a complete set, and it comes down as something you can open in Excel. And you can also, actually let me go back one, there's also a button for identifiers not found, which I think is an important part of the analysis results. So if you submit a set of identifiers and some of them don't match anything in Reactome, it's very important that you know that, because those identifiers might actually represent a process, but they're simply a pathway process we don't have in Reactome. So you need to know that your match can only ever be partial. So it's something very worthwhile taking note of. Okay. The other kind of analysis you can do is the expression overlay analysis. So this here, it was intended to be used with microarray data, but it will work with essentially anything where you have a molecular identifier associated with a number. So you can also use it for genotyping results if they have a score. But here I have a microarray experiment where I've submitted using probe IDs and several different time points. So the numbers here are showing expression at a series of time points. So this tool is great for doing a time series or a disease progression, something like that. And when the results come back, it's shown to you as colored overlays on top of the standard pathways, and the color will represent the level of expression, which is indicated by an expression bar on the right hand side. For a complex like this object here, you have bands of color because there are some objects have a higher level of expression than others, and then some objects within this complex had no expression data available. So probably this complex contains some small molecules that weren't represented by the microarray data. And because I submitted multiple numeric columns, I have a toolbar here which allows me to step through the columns of data, and that will be revealed as changes in the overlay color as I click on the button, which allows you to then visualize how the expression is changing through the samples you submitted. Yes. The last tool then is the species comparison tool, and that's very simple. You're always comparing human to another species, so all you do is select the species you're interested in from the drop-down list. And when you get, again, it's shown to you as overlays on the standard diagrams, and here the yellow coloring is showing you that the ortholog was identified. So here, any object in yellow shows you that that molecule can be identified in the species that you selected. Anything that has no overlay, we weren't able to identify. So, and that will, for small molecules, they will never be predicted to have an ortholog because we regard small molecules as being species agnostic. The molecular interactions overlay I really already showed you, but the thing to, I'll zoom on a little bit to show you this settings panel. So within that settings panel on the right hand side, the default database is intact, and those interactors are shown as the red circles that are superimposed onto objects and will appear very fast when you click on them. But you can actually change the database. You can select any database that's available as part of a service that's called Psychic, PSIC Quick. So you can select from any of this list of other alternative databases, and then the information will be fetched back and become available for the diagram. And you can see here I've done that for the Chembl database, which is now allowing me to show chemical interactions with my selected protein molecule. Okay, so thank you for bearing with me. I ran over time a little bit. If you want more information about Reactome, there's a user guide document on our homepage. You can contact us at help@Reactome. There's a very nice video tour built into the Pathway browser, and there are a couple of other EBI training experiences that you can try out as well. So thank you.